Hello, everyone, and welcome back. This is a continuation of our discussion of diseases. Chapter 24 will cover the digestive system infections. So, we'll need to talk about the digestive system basics first. This is all about the gastrointestinal tract. That's the main pipeline: the mouth, the throat, esophagus, stomach, intestines. It's basically, you can think of it as one long tube handling what you eat. Then you've got accessory structures like the teeth, tongue, liver, gallbladder, and pancreas. These aren't in the tube, but team up with it. The teeth and tongue break down food mechanically, while the liver, the gallbladder, and the pancreas, they produce chemicals like bile and enzymes in order to help break down food into molecules small enough to absorb, which then gets sent to your bloodstream, your lymphatic system, so on and so forth, and keeps things going. So let's move to the normal microbiota of the digestive system. Most sections are actually heavily colonized. Approximately millions of bacteria per milliliter of saliva in the mouth. And in the large intestines, it's about 100 billion cells per gram of feces. The stomach and the small intestine, by contrast, have far fewer microbes due to the harsher conditions in those environments. And overall, these microorganisms play an important role in the digestive tract. They assist in degrading complex molecules. They also, as a byproduct, produce essential vitamins and prevent colonization by transient or opportunistic pathogens, which is a critical microbial community process in order to maintain a healthy digestive system. So, as in our fashion, we'll be grouping these based upon the types of organisms. We're going to talk about bacteria and viruses, namely. We focused more on the eukaryotes that infect humans on chapter 5, the eukaryotes of microbiology. So, we're going to just focus on bacteria and viruses here. In archaea, as we know, there's no strong evidence that suggests they actually cause disease, although there's some correlation. So far, we don't have strong evidence that they're causing diseases. And we've split the bacteria into two separate groups: those that are involved in the mouth, which may be dental caries, periodontal disease, and those that are involved in the bacterial lower digestive system. So these are any number of enteric bacteria and others. Now we're going to start at the top here with the bacterial mouth organisms. So we'll cover dental caries or tooth decay. Teeth aren't living cells for the most of it anyway. There are parts of it that are living, but the outside of it is easily inhabited by living cells, microbes, that can form what we call dental plaque. The decay happens when bacteria, and this is namely, it's going to be due to Streptococcus mutans, which is shown in figure A and figure B. They are capable of metabolizing sucrose and carbs, and they're very efficient at this. And they, instead of producing a lot of alcohols like maybe some other dental microbes, they prefer to produce a lot of acid. And that acid is what's actually decaying and eating away at our enamel. So S mutans actually really thrives in our mouths, in particular with the current diets that we consume. It handles a wide range of sugars as well, as that's its specialty, and it tolerates very acidic conditions. And it also produces dextran, and that's the sticky stuff and plaque that allows it to adhere tightly to our teeth. Moving along, we get the diseases here from all this. One is periodontal disease, which is a broad term for issues with the structures supporting the teeth. like the gums. It starts with gingivitis, which is infection and inflammation of the gum line. And this is affecting about 50% of US adults. And I wouldn't be surprised if that number is climbing. If that becomes a chronic problem, it can turn into a more serious disease called periodontitis. That's when you get periodontal pockets with pus, and it can damage the root tips of your teeth, eventually leading to tooth loss. It's a progressive degeneration, and it's moving from gum irritation to serious structural breakdown over time. Okay, so that was pretty short. We're going to spend most of our time settling in here talking about the bacteria of the lower digestive tract. And before we do, we need to be able to differentiate between two different types of diseases or issues that are caused from these bacteria, and that is infections and intoxications. Now, sometimes we lump these together and we say we've had food poisoning, and that's more of a day-to-day colloquial term that we ate something and there was something that didn't agree with us in that food. But infections and intoxications have specific meanings in microbiology and in health. So, infections is what we mostly talk about when we refer to bacteria infecting. So, you ingest a pathogen, and that pathogen multiplies in the GI tract and infects the tissues there and causes damage. That's going to be characterized by a usually, I mean, as a general rule, it's going to be a delay in symptoms because the bacteria needs to multiply and it needs to, or it might need to spread as well and invade tissues in the surrounding area. So this takes more time, and typically it will come with a fever. And a fever is, again, your way of your immune system of trying to fight off an infection. So it can come with that systemic fever. Whereas intoxications, these are caused by ingestions of, well, toxins that are made by bacteria, and that impairs or affects the GI tract. And these symptoms appear more suddenly. They can include diarrhea, cramps, and vomiting. Fever can occur if it's a really high dose and a really toxic toxin, but it's less common. Usually, these are more localized and less systemic. And um, a localized issue is going to be more about inflammation than about having a fever. Okay, let's first talk about staphylococcal food poisoning. This is where you get intoxication by ingestion of Staphylococcus aureus enterotoxins. And it takes the following progression. So let's walk through the storyline here. So you have food, it needs to contain some protein in it. That's what Staphylococcus aureus likes. And it gets cooked, and that usually kills all the bacteria. And I want you to imagine this is kind of synonymous with how superinfections start, right? We've cleared the playing field of bacteria, and superinfections is with antibiotics. Here we're killing them through cooking. And then that food is contaminated by a worker. So we carry the Staphylococcus aureus on our hands. And so they introduce that into the food, and there's no competition there. All the other bacteria has been eliminated. And if we don't keep the temperature if we don't keep the food at the right temperature, either it needs to be hot enough that the bacterium is outside of its growing range, or it needs to be kept very cold so that it's outside of the growing range. If it's somewhere in that sweet spot, in the minimal and maximal temperature, that organism starts to grow. And if it occurs long enough and you get a buildup of enough organisms, you're going to get a release of toxins in a high enough amount that it's going to cause an intoxication if someone consumes it. And what can also occur here is these toxins can be something we call heat labile. And that means that they might not be affected by reheating. Heat labile means they are affected. If they're not heat labile, then they're unaffected. And so you might think, okay, we've had a soup that's been sitting out for a while all day long at a party. I'll throw it in the fridge the next day. I will just microwave it really well or heat it up on the range really well. And you may kill the staphylococci, but the toxins remain unharmed. That food is then eaten, and in a short amount of time, here we have 1 to 6 hours, intoxication occurs. It doesn't take very many bacteria. As I discussed, in the human tract, digestive tract and feces, there's about 100 billion bacteria in 1 gram. And to put that in perspective, 1 gram, there's about 1,000 grams in a liter of water. So if you have a soup, you can multiply that by 1,000 pretty easily if it grows up to a high enough concentration. It doesn't take anywhere near that amount to create a intoxication. It only requires about 1 million bacteria, so 1/1000th the concentration per gram, and that's enough to elicit intoxication. And what we recommend, the best preventative measure, is to just not allow it to form. So keep your foods either cold or keep them hot. And that's really important for food safety. If you go to a buffet or something, they have to keep those soups at a certain temperature. If it's too cold, then you're going to have the microbes grow. Let's move on to shigellosis. And this is also called bacillary dysentery. It's an infection from 4 different Shigella species, and they multiply in the small intestine over 1 to 3 days before symptoms hit. Now, the most common one is from Shigella sonii, and it tends to have milder effects. However, one that is a little bit less common, but globally dominates in developing areas, is Shigella dysenteriae. You can imagine what this causes. It has severe symptoms, and that's thanks to something called the Shiga toxin, easy to remember. And you can think of having a strong fever, cramps. It can cause up to 20 bowel movements a day. Worldwide, estimates are that it causes over 500,000 cases, although it's hard to know because they're not all reported. And mortality can reach up to 20%, which is incredible. What kills people from this disease is typically dehydration. So water and electrolytes could be a lifesaver here. Now let's go on to salmonellosis. This is caused by non-typhoidal Salmonella species. So not subspecies typhi, essentially. There are several species that can do it. It's mainly S. enterica, which is the most common one, but sometimes S. bongeri can cause it as well. These bacteria pass through the intestinal mucosa, so they can spread into the cardiovascular system and the lymphatic systems, and that makes it a systemic issue. Again, we're looking at an infection, so it's not going to be like 1 hour and you start feeling awful. If we're to diagnose someone, we're looking for a much longer period as it spreads throughout the body. 12 hours is kind of on the short range of things, up to about 36 hours. And what we see is typically a moderate fever, cramps, diarrhea, and nausea. In the US, it's estimated that over 1 million cases occur yearly, with greater than 400 deaths, which is pretty significant in the US for foodborne illness, although the number of cases could be higher because not everyone goes in to seek medical attention for these. All right, so let's talk about the typhoidal variety. This is caused by a specific cerevar. So we have the species the genus species, which is Salmonella enterica, and a cerevar is like a certain strain of it, just like you might get heirloom tomatoes, which are they're all in the same species, but you get specific substrains, and that's something we sometimes call subspecies or cerevars. So we have our Cerevar typhi, sometimes abbreviated just as S. typhi, and it spreads through the what we call the fecal oral route. And you might hear that, and you think, that sounds disgusting. But the fact of the matter is, um, microbes that come from fecal matter are just everywhere. They're all over your phone. They're all over your hands. They get on your body, on the doorknobs. They're just all over the place. It just happens. You flush the toilet, and even just the droplets that form from the toilet spread it all over the bathroom. But this particular disease, it's pretty rare in the U.S. now, and that's thanks to our sewage treatment infrastructure. The bacteria multiply actually in phagocytic cells, and they spread through our different organ systems. After about a 2 to 3 week incubation, you'll see headaches, fevers, and later diarrhea. Recovery typically gives lifelong immunity, but the catch here is that about 1 to 3 people become chronic asymptomatic carriers. Asymptomatic symptoms mean that you're showing signs or symptoms of the disease, and A means without, so they're just carrying it, but they're not exhibiting any signs. But they continue shedding the pathogen long term. A famous example, as you can see in the image to the right, is Typhoid Mary. Her name was Mary Malone. She was a big new sensation um, in the early 1900s because she was a cook, a family cook. That's what she did for a living. And she was an asymptomatic carrier. And she knew that she was a carrier. It had been identified. People had died from her getting others infected. And she just kept moving around and picking up new work and infecting other people. And this infected many people throughout New York. So she refused to stop working despite spreading the disease. Public health officials eventually quarantined her for life. And that really highlights how carriers can drive outbreaks, but also the degree of actions that the government has deemed appropriate to take in their intention to combat the spread of disease, which is pretty pertinent in today's society, as we've had our own recent pandemics, and there is, a history of the government taking these sorts of strong responses in the past. So let's talk cholera. This is caused by Vibrio cholerae, and this is a curved rod with one flagellum, as you can see in the image on the right. It's endemic in coastal waters. It's just growing in biofilms on crustaceans, so on and so forth, which is a reason why I will not eat raw oysters. I don't care how much you pay me. Also, I don't really like the taste of it. Um, Once ingested, it colonizes the small intestine and it releases an exotoxin. And that exotoxin pulls water and electrolytes out of your host cells. So it's like a diuretic. It's drawing that fluid into your intestine, and it can lead to an incredible amount of fluid loss. You know how it gets out. Up to 5 gallons of fluid can be lost a day. And when you're having that sort of a violent bowel movements, this can lead to bleeding, and it can lead to shock and death. Um, and additionally, just the dehydration is a big issue. So, it's very important if you're losing that much fluid, you need to be bringing in that much fluid, plus the electrolytes that come out with it. So, now with treatment, we can drop mortality rates down to about 1%, right? But without treatment, mortality is up to about 50%, which is just wild. Now we'll talk about E. coli gastroenteritis. This seems out of place as it is the most common bacterium in our gastrointestinal tract. It's normally harmless. E. coli can turn virulent through gene transfer or lysogeny, which we would then call that bacterium enterotoxigenic. So it's an enteric bacterium able of creating a toxin. We've got different types here. So we have enteropathogenic. This destroys intestinal microvilli, and it secretes proteins that cause diarrhea. Enteroinvasive infects epithelial cells, triggering systemic illness. Enteroaggregative means that we have clumps around cells that form, and this is associated with secreting a diarrhea-causing toxin. And lastly, we have enterohemorrhagic pumps. Ouch. These are a Shiga-like toxin, and it causes the bloody colon issues or kidney damage that we've discussed before. Often cattle can be a reservoir for this, and enterotoxigenic E. coli is the top culprit for travelers' diarrhea. Now let's cover campylobacteriosis. This is caused mainly by Campylobacter jejuni. It's a gram-negative microaerophilic spiral. It's the most common bacterial gut infection, with poultry and cattle as reservoirs. Virtually all poultry meat and 60% of milk carry it, which is why we generally pasteurize our milk. Symptoms include fever, cramping, and diarrhea. Usually clears up in about a week. Now let's cover peptic ulcer disease. This is caused by Helicobacter pylori, which is a microaerophilic spiral bacterium. If I might just interrupt here, this bacterium, it causes ulcers in the stomach or the duodenum. And for a long time, scientists supposed that ulcers were just really caused from too much stress. But as we discovered, there was a bacterial link, and there was a scientist who had hypothesized that it was caused from this bacteria, and he was kind of laughed at and disregarded. And so, what did he do? He took Koch's postulates, and he decided he would harvest this from a sick patient. He would grow it up. He would give it to himself, a healthy person, and he developed the symptoms. And then he treated himself with antibiotics, and the symptoms went away, which was a pretty revolutionary thing to do for the time, exposing yourself and experimenting on yourself like that. But he made some groundbreaking discoveries. It infects an incredible 30 to, so I've seen estimates of up to 50% of the developed world. 15% of them develop ulcers of stomach and duodenum. And the way it works is it disrupts the mucus layer that protects the stomach lining from, or the protecting stomach lining gets degraded, and therefore your stomach gets exposed to those acids and enzymes inside of it, and those lead to the formation of sores or ulcers. And again, usually antibiotics are pretty good at clearing this up. So moving along, let's talk about Yersinia gastroenteritis, or yersiniosis, as it's sometimes referred to. This is a bacterial infection that affects the gastrointestinal tract. The illness is caused by two species of Yersinia. We have Enterocolitica and pseudotuberculosis. Both are gram-negative rod-shaped bacteria that can survive and grow in cold environments, which is important when considering food safety, as they can grow in foods that are refrigerated. The primary reservoir for these bacteria is livestock, particularly pigs. Humans typically get infected by consuming undercooked or contaminated meat, especially pork. What makes Yersinia unique is that it can invade tissues, targeting the intestinal lining and sometimes spreading to the lymph nodes. So, symptoms, as you can imagine, would include fever, diarrhea, which may be bloody in several cases, and right lower quadrant pain, which can mimic appendicitis. This is especially common in children and young adults. The illness usually lasts 1 to 3 weeks, though in some cases symptoms can persist longer or lead to complications like a reactive arthritis that can occur. So understanding Yersinia is important both for the clinical diagnosis side and foodborne illness prevention, especially if you don't want to misdiagnose this as appendicitis or something like that. Particularly when we're dealing with undercooked meat and proper food handling procedures. Clostridium perfringens gastroenteritis is a common cause of foodborne illness. Typically, it's linked to meat dishes that have been improperly cooked or stored. The bacteria is gram-positive, rod-shaped, and capable of forming heat-resistant endospores. This means they can survive cooking, then germinate, and multiply if food is left at unsafe temperatures. Infection happens when people consume meat contamination with animal intestinal contents. And once inside the intestines, the bacteria release an exotoxin, which disrupts the fluid balancing and leads to symptoms. These symptoms include watery diarrhea, abdominal cramps, but typically no fever here, since the illness is caused by a toxin rather than an invasion or infection. Fortunately, it's typically short-lived. Symptoms usually resolve in about 24 hours, though dehydration can be a concern in severe cases. Prevention here just comes down to proper food handling, keeping cooked meat hot or refrigerating it quickly to prevent bacterial growth. Next up is Bacillus cereus gastroenteritis. Bacillus cereus gastroenteritis comes from, it's a gram-positive rod. Sometimes it can be called gram variable, and depending on the growing conditions, its cell wall, even though it's always gram positive, but the color that it exhibits can sometimes look more gram negative. So, depending on its growing conditions, it might affect how well it takes up and retains the stain. But the structure is like a gram-positive organism, right? You have the plasma membrane and the outer cell wall. It doesn't have an outer membrane. It's a normally harmless species. It's found all over in the soil and on vegetation. You grow it all the time if you go out and take a soil sample and streak it out. Where we get into trouble though is that it makes endospores, which we know are heat resistant to just boiling temperatures. And those endospores, after cooking food, then it can germinate in the food. And again, we have no competition, and that means we have high numbers. Additionally, those exotoxins, if you have a gastroenteritis version of Bacillus cereus, can build up and survive reheating. So it's not heat labile. Symptoms include vomiting, diarrhea, or both. You can have a rapid onset that lasts less than 12 hours, depending on how much bacteria and exotoxins have built up. Next, we have Clostridium difficile associated diarrhea. This bacterium actually is normally a harmless gut resident, but it can overgrow as a superinfection where antibiotic therapy, the broad spectrum antibiotic, can wipe out competing microbial flora. And that leads to symptoms that range from mild diarrhea to severe colon inflammation, and this one can especially be deadly. Treatments usually involve antibiotics, or in tough cases where they might be antibiotic resistant, actually a fecal transplant, where, as you can see in this image here, these are the suppositories, and you can take a fecal sample. It's usually from a relative who's healthy, and you basically clean up that fecal matter and spin down and harvest only the cells. Those cells are then used as a suppository in the lower colon and that helps to restore the microbial, the normal microbiota. And this cartoon shows the progression of the disease. If we start over here to the left, we can see that the application of a broad spectrum antibiotic, here's a variety of them, you don't have to memorize those. They knock out the normal microbiome of the gut. And the C. difficile then can, first it forms endospores that can survive the acidity of the stomach and end up down in the large intestine. These C. difficile then germinate and they wreak havoc in the gut and flourish there. They release toxins. These toxins can actually damage the mucosal membrane here, and this can cause severe diarrhea. And because this is associated with broad spectrum antibiotic treatment, this is actually the most common cause of nosocomial diarrhea. Again, that's hospital caused infections or infections that occur as a result of being in the hospital. Okay, so that wraps up our discussion of bacteria. Let's go ahead and switch bases now and talk about some viruses. Now, I did want to just touch upon mumps here. Mumps is a virus. It colonizes the parotid glands. These are the salivary glands that are below and in front of the ears. And as you can see in this child here, especially in the far right of this picture, his left side, you can really see that swelling taking place there. It's transmitted in saliva and respiratory secretions. And it can also cause inflammation in men in the testicular region, and a rare complication can be sterility. So it's important to take care of this and treat it. And it's you can protect against it through just the MMR vaccination. We're going to talk about hepatitis. There's 5 different viruses clinically associated with hepatitis. This is the inflammation of the liver, and it's caused by one of at least 5 viruses. I just tried to simplify things here into this table, which focuses on the highlights. And we can go through one by one here, starting at the top in alphabetical order. So hepatitis A, it's transmitted through the fecal oral route, and it's mostly subclinical, leads to some appetite, nausea, diarrhea, malaise. Some jaundice can occur naturally if you're affecting the liver here. It's not a chronic infection though, and there's a vaccine for it as well. So the amount of hepatitis A in the population has gone down greatly. Hepatitis B is more serious. It can be transmitted it can be transmitted sexually through the parenteral route, which is if you get a cut, even through the perinatal route, so transmission of an infection from mother to baby around the time of birth. It's similar to HIV for most of our symptoms here, but it can cause a chronic infection, and it can actually lead to liver cancer or cirrhosis of the liver. Again, with HIV, HBV has a vaccine available, although there's estimated to be a lot of chronic carriers worldwide, still about 350 million, so that's a little bit less than the US population. Hepatitis C is some serious business. It's transmitted through the parenteral route, and it's similar to HIV, but it takes a long time. It has a very long latency period, about 20 years for the appearance and recognition of symptoms. And it can be chronic. About 85% of the cases become chronic. It actually kills more people in the US than HIV does, and there's no vaccine. Hep C, the reason why there's no vaccine, too, if I might just say, it's highly genetic variable. There's 7 major types, and you could be reinfected with them. It only infects humans as well, so it's difficult to start off with animal models in order to study this virus. Hepatitis D, again, it's the parental route, and it's typically after a chronic Hep B infection that this, it's going to be after a Hep B infection. It kind of piggybacks off of hepatitis B. It causes a severe liver damage and has a high mortality rate. But because there's a hepatitis B vaccine, we're not so worried about developing a vaccine for hepatitis D because one comes before the other. It needs a helper virus. Hepatitis E, it's ingested, so the fecal oral route, similar to HAV for infections. It's not a chronic. For symptoms, it's also not a chronic infection, but it does have a particularly high mortality rate, greater than 20% in pregnant women who have a suppressed immune system. Let's shift to viral gastroenteritis now. First, we'll talk about rotavirus. Rotavirus brings a low-grade fever, diarrhea, and vomiting. Lasts for about 1 week. In the U.S., there's a lot of cases, 2.5 million cases yearly, and there was a vaccine in about 2006, which has actually reduced the cases by about 98%, and deaths are now under about 100 per year. Norovirus is another one of those that you might hear a lot occurring on cruise ships. It causes vomiting or diarrhea that lasts for about 2 to 3 days, but it's pretty bad for those 2 to 3 days. There are about 20 million cases annually in the US alone, and greater than 800 deaths. And it spreads very easily. It's tough to kill, and it lingers on surfaces for a while. And that's why in places like cruise ships, this stuff just spreads like fire throughout the whole crew. And that wraps up our discussion. It was kind of a short one here. Hopefully, you learned a lot about these diseases, and we'll see you on the next one.